All right, let's go ahead and get started. I'm going to finish off chapter 8 today, but straight to your minds from mine. This is where it's going to stop. So the exam's going to stop right here at translocations. All right? So we started talking about aneuploidy last time, but right here at translocations, slide 32 is where the exam is going to stop. So we talked about this a little bit in lab. You guys did non-disjunction. You can get out your chromosome bags at home and do this again if you want to review. They're actually very helpful in that way. So non-disjunction during gametogenesis. So if you have non-disjunction during meiosis 1, there will be no gametes for that given chromosome that are normal, because the homologous chromosomes, which are supposed to separate from each other at anaphase of meiosis 1, do not do so. Both go to one daughter cell. So right here, none of the cells after meiosis 1 are normal. And then here you have nothing. Here you're going to have, because in meiosis 2, you don't have chromosomes pair anymore. You just have the individual chromosomes line up at the metaphase plate, and then the sister chromatids separate. Each of these two chromosomes will become two chromosomes, because as soon as the sister chromatids separate, centromeres are separate, they count as individual chromosomes, and you get two. The blue one here is the homologue of the red one. That's the way the book is writing it. So here, these gametes don't have any of the chromosome in question. They get one from the normal gamete, and so they're monosomic for that chromosome. Whereas these have 2 plus 1, and they are trisomic. Again, 2n plus 1 means that you have 2n, two complete haploid sets, and then one of one additional chromosome. It's really important here to remember that all we're showing is this one homologous pair out of all the chromosomes that are present. So these gametes would be normal and have one each of all of the other chromosomes that they're supposed to have, that have one complete haploid set. And these zygotes here would be diploid for everything except the one chromosome that we're talking about. All right. Non-disjunction during meiosis 2. We're assuming here that meiosis 1 happens normally, so the two homologous chromosomes separate into daughter cells. And then, because non-disjunction is rare, if non-disjunction happens in this cell during meiosis 2, leading to one gamete with two chromosomes and the other one with none, this cell is going to not suffer non-disjunction for that chromosome, and you get normal. So if you have non-disjunction during meiosis 2, you'll get a 50-50 normal gametes versus aneuploid gametes. And the reason for that is, again, because non-disjunction is such a rare event, that yes, it's plausible that non-disjunction that's happening here could also happen with this cell for the exact same chromosome, but it's not probable. It's such a rare event. So then these two cells would have 1, and plus 1 is your normal diploid cell. And here again you would get one trisomic and one monosomic. 2 plus 1 is 3. 0 plus 1 is 1. Talked about this last time. 2, non-disjunction during mitosis. So non-disjunction can happen during mitosis, but unlike non-disjunction in meiosis here, so in either meiosis 1 or meiosis 2, the cell that is affected, here a trisomic cell and a monosomic cell, those are the zygotes. So every cell in that organism will have that same defect. when you get a non-disjunction during mitosis, it only affects the progeny or the clone of cells that come from that one cell that had the problem. If it happens very early during development, then it will be a lot of cells that have the problem. If it happens relatively later, it could be very few. And I said this, so problems with aneuploidy lead to cancers, and so that, you know, that would be development of a cancer in some specific one cell, and then that develops into a tumor later. And in most cases, that doesn't even happen, because if you get aneuploidy, in a lot of cases the cell can sense that something is wrong. Your aneuploidy, in a lot of cases, will lead to dysregulation of mitosis. So instead of the cell stop dividing, it will continuously divide, and the cell will usually recognize that, engage apoptosis, and the cell will be dead, and that's the end of it. So you have to have multiple things go wrong there. All right, and so you can get, in this case, every cell that comes from this one will be monosomic. Every cell that comes from this one will be trisomic. And in just a second, we're going to talk about Down syndrome, but I wanted to mention again that I had a student once that, in addition to the two ways of getting Down syndrome that we're going to talk about coming up, he had a brother who had Down syndrome by this method, so he was a mosaic for it. They did genetic tests with multiple tissues, I guess, and found that some of his cells were, well, there's no cell that's normal here, but some of his cells were trisomic that had 3 copies of chromosome 21, and some of his cells were normal 2N and just had 2 copies. So I assume in this case with Down syndrome that the monosomy is not viable. You know, the cell just undergoes apoptosis and dies, but the trisomy is fine. So he had, many of his cells in his body had 3 copies of 21, and many others had just 2 like they should. All right, so we talked about translocations. The translocations we talked about before were reciprocal translocations. So remember, there was the blue chromosomes and the red chromosomes, and a piece of one of the blue ones and red ones broke off, and they got switched and went back on the opposite chromosomes. There's also non-reciprocal translocations. If I didn't mention that before, sometimes you can get a piece of one chromosome breaks off and just gets added to a different chromosome, and that other chromosome doesn't donate any piece back to the first one. A third way that you can get translocations is this way. So a Robertsonian translocation is when you actually get breakage resulting in 2 smaller chromosomes combining to make one large chromosome. So if you have a break here and a break here, and there's not too much on the ends that's important, you know, these are going to be acrocentric or telocentric chromosomes where the centromere is very close to the end, you can actually get one either metacentric or submetacentric chromosome where the centromere is either right in the middle or just off to one side, and a little fragment that as indicated here usually gets lost. And so if you came to the review session, you know what I'm going to say next. I forgot to mention, if you didn't make it to the review session yesterday, it's recorded and it's up online, so everything is online now for you to look at for the exam on Thursday. And I'll post this lecture right after today, but like I said at the outset, we're only going to that slide with the translocation, so if I don't get this posted until tomorrow, it's no big deal because none of this is going to be on there. All right, if you look at the other great apes that are not us, chimps, which are our closest relative, so 99 point something % of their DNA and our DNA is identical in sequence, or gorillas or orangutans, these are 2n equals 48, not 2n equals 46 like us. So their haploid number, instead of being 23 like ours, is 24. And the reason is because where we have chromosome 2, they have two different chromosomes, and you can see the banding on here kind of matches up. The left-hand side of human chromosome 2 matches one of these chromosomes, and the other side of chromosome 2 matches the other ones. This is kind of an old-timey slide where you used to do banding of chromosomes to see what various stains would make light and dark marks on the chromosomes there. But if you sequence these, you can also see that all the genes that are here, and all the genes that are here are on that one. So sometime after the fork occurred in which humans and the great apes last shared a common ancestor, we have had a Robertsonian translocation that took these two chromosomes and made one out of them. So after we shared the common ancestor, the lineage that led to humans had the Robertsonian translocation where all the other great apes did not, and so they still have two chromosomes there. So essentially the same content, it's just in different chromosomal carriers. All right, so most aneuploidy is lethal. If you go back to, I would get out or download again your lab number 1, and if you look in the introduction, there is a chart in that is really interesting. So they took samples from a bunch of random healthy live births and did a chromosome smear on them, and they also took samples from a whole bunch of spontaneous miscarriages, so fetuses that on their own failed to develop. And you will look and see that in the spontaneous miscarriages there are a whole lot of aneuploidies. In the live births there are not very many aneuploidies because most aneuploidies for humans are lethal, meaning at some point during development you're done, with two exceptions. Like we talked about earlier, the sex chromosomes, except for SRY gene, there's not very much information on the Y. I don't know that there are any other genes that really do anything. There probably are, and I'm just not aware of them. And although the X chromosome has many genes that are useful and in fact are required for development, so you die if you don't have an X chromosome very early on. Extra X's are shut down by dosage compensation that we talked about. And so it's not, the rule is, it's not one X gets shut down so the other X can be active in females. The rule is only one X can be on and the rest get shut off. So if you have extra X chromosomes, they all get shut down. And the only problem that's left from that is that you have those genes that are out toward the pseudoautosomal regions at the end that stay active and can cause some issues. And this is what we're talking about. Why Down syndrome is which chromosome? 21, right? And how many chromosomes do we have? 23, but 22 autosomes, right? So if I didn't say it at the beginning, I think I say it when I show that picture, chromosome numbers are always listed from the largest to the smallest. So chromosome 21 is the second to the smallest. But even given that, you don't see any aneuploidy with chromosome 22 because having 3 or 1 for even 22, the smallest one, is usually lethal. And I bet if you look in that chart, you will see that in the spontaneous miscarriages, you will see some aneuploidy in chromosome 22, but not really in the live births. All right, so 3 forms, right? We already talked about the mosaic Down syndrome, but we're going to talk about primary and familial. So primary Down syndrome results from a non-disjunction event that occurs in the mother. This one does not run in families because it is spontaneous. It's a rare miscarriage event it's a rare non-disjunction event that occurs. And so you can see, now you can look and see the size of the chromosomes goes down, but even though 22 is smaller than 21, just 2. 3 is no good and 1's no good. So now we have 3 chromosome 21s here. As it's discussed in the lab handout, and I think I talked about with you guys in the lab, primary Down syndrome usually, but not always, results from non-disjunction at meiosis 1 and in the mother, not the father. So the reason for this is because most of the oocytes that a female human is ever going to have are locked into prophase of meiosis 1 at the time you hit puberty or shortly thereafter. And so then one develops every 28 days, usually from one ovary, then the next, and back and forth until menopause occurs. But you're just getting an egg that's already at the starting gate and gets going. So the longer it is before childbearing, the longer the egg has been hanging around, and the longer time there is for something funny to happen with that spindle that's already been set up. So if you look here, here's maternal age and number of Down syndrome per 1,000 live births. It's kind of a very low level until, you know, right here. And then between age 40 and age 50, it's like, so between 30 and 40, you go from 1 in 900, or not 1 in 900 to 1 in 100, and then 1 in 12. Because the oocytes have been sitting around so long that there's a lot that can happen. And so in many cases, as maternal age increases, sometimes it will be suggested that in vitro fertilization be done so that eggs can be extracted and examined so that before implantation you can see that everything is okay and you don't get one with a spindle that has defects. Because in addition to Down syndrome, which is survivable, you also get an awful lot more aneuploidies that aren't survivable. And if you look in that chart that is in the handout, one of the interesting things in there is you see a lot of chromosome 21s and the chromosome 21 trisomies in the live births, but you see even more in the spontaneous miscarriages. So that children that are born and have Down syndrome are the lucky ones. I believe it's like 2/3 don't get to be born. So it depends on what you get for the other chromosomes. So trisomy of 21 is survivable, but the rest of your genome matters as well. See what you get there. All right. So familial Down syndrome, where it runs in families, is actually the result of a Robertsonian translocation, almost always between chromosomes 14 and 21. And so in a normal individual, here's chromosome 14, here's chromosome 21. The gamete will have one of each. But sometimes you can get a translocation and you will have an individual who's a male or a female, because these are both autosomes, neither is the X or the Y, that will have this genotype. Instead of having 2 14s and 2 21s, they will have 1 14 and 1 21, and then a third chromosome that is the 14 and 21 Robertsonian translocation. This carrier will be phenotypically normal, and for most, I would say in all cases, would never know that they're a carrier unless they have genetic testing done. Because even though you have 14 and 21 stuck together here, you still have the equivalent of 2 21s, this and that half, or that piece, and two 14s, one regular 14 and one piece right there. Problems occur, however, during gametogenesis, because unlike over here where the two 21s compare and the two 14s compare like they should, now you have just three. And so here's what can happen. So either you can get the two 14s pair and the 21 is separate, so that means the 14s will go to separate, to separate cells here, and then the 21 goes to one of them. Or you can have this case in which the two 14s pair again, but the 21 goes here. Or you can have case C on the right where the two 21s pair and switch, and then the two 14s go together. In this case, part A, you have what will be phenotypically normal individuals. Because you get one 21 and one 14 here, when this gets fertilized by a normal gamete, either a sperm or an egg, depending on what this is, you'll get another copy of 14 and 21. So this individual will be normal, and that's it. The familial Down syndrome is done for that entire lineage. Here, you will have a translocation carrier exactly like this person up here. This person will not have Down syndrome, but will have the capability of passing the translocation on to their offspring or having Down's offspring. This is the only case of everything else in which you have a live birth and you end up with Down syndrome here. So you have one normal chromosome 14, one normal chromosome, well actually a piece of what would be chromosome 14, one normal 21, and then the other piece of 21 that's here. So when you get another 14 and another 21, you'll have two 21s plus this, so that's 3. And then the normal 14 goes with the other half of the translocation. So really you are diploid and not aneuploid for 14 here, but you are aneuploid for 21, and these individuals will have Down syndrome. So even though trisomy 21 is tolerated to some extent, and again, a lot of trisomy 21s don't survive gestation, monosomy of 21 is not tolerated. So these individuals will result in a spontaneous miscarriage at some point during development. Likewise with trisomy 14 or monosomy 14. Right? So this one is going to have two 14s here. The resulting zygote will have three 14s because you have the translocated 14, a normal 14 here, and then this one comes from this 14 and this 21 come from the other zygote. You're okay for 21. You're actually diploid for 21 because there's one and here's one. This guy over here is okay for chromosome 21 because you get one like you're supposed to, but it got no 14, and so you're monosomic for 14, and that's not viable either. So these families, in addition to having Down syndrome, this individual, in addition to having Down syndrome in their progeny at a much higher than normal rate, because you have 1/3 of live births are going to be trisomic here versus a very rare event in the case of non-disjunction, you are going to have a lot of miscarriages. So even if you don't, even before you have a trisomy 21, if you're trying and trying and trying, and your obstetrician determines that you're pregnant, and then you keep losing again and again and again your pregnancy, a lot of times they will have your karyotype done, because in addition to this, you could also have something else going on that's leading to these spontaneous miscarriages. All right. Uniparental disomy. So we haven't talked about disomy yet, right? So monosomy is having one of a certain chromosome, trisomy is having two, tetrasomy is having three, or having four. We haven't talked about disomy yet because that's just having two chromosomes. That's being diploid. That's normal. But in this case, what's not normal is you're going to be having two copies of a certain chromosome, but they're both going to be from the same parent. And this starts off with, and I've put the female gamete here, but it can be either. You will get non-disjunction during meiosis, which will lead to a gamete that has two of a certain chromosome. And remember, in most cases, this is going to be lethal, and you won't get development because 3 like this is not going to be tolerated. However, you can have two weird things happen. So the first thing is this happens in one of the gametogenesis. You get a non-disjunction event. And then after you're a zygote, but before you reach the point at which failure to develop occurs, you lose loss of one of the chromosomes. Now you could lose either the red one here or one of the blue ones, but in this case you've lost the red one, which is from the dad. So you end up with an individual that for this particular chromosome, both of them came from one parent. This can be a weird thing when you're doing like paternity testing. If you use a marker for paternity testing from this particular chromosome, it will look like nobody's the father, right? It would look like parthenogenesis, right? Like the mother just had a child and there's no other, there's no other markers there. Or that the mother, that the father had the exact same markers. But if you look at the DNA sequence on other chromosomes, then it would be fairly obvious what happened there. So some organisms like C. elegans, once the egg and sperm come together to form a zygote, at that very first cell division, it's stereotypical development. The first cell divides into these 2 cells, then they divide into 4, but every cell has already had its fate specified. There is a lineage of cells. Mammals have a different form of development. So one cell divides to 2 to 4 to 8 to 16, and sometime, I think it's between 16 and 32 cells, every cell in that developing organism could make the entire organism still. And so if you got death of some of the cells, it would be fine as long as other ones live. And so the cells that maintain the 3 chromosomes would all be dead, and the individual would be made up entirely of the cell that lost the paternal chromosome there. That's why when you're making transgenic mice, for example, you can take embryonic stem cells from one mouse and inject them into the blastocyst or growing ball of cells from a different mouse, and they will colonize that and become part of the organism because the cells are not specified to a certain fate yet at that time. All right. Another weird thing that occurs, and you guys have probably not seen a fruit fly that looks like this, but you have seen the pictures of like the cardinal that's like half the red cardinal and half the brown cardinal, like half the male and female coloring. You can get what are called gonadromorphs here. And so in fruit flies, remember that sex is determined by counting X chromosomes. The Y chromosome is present in males, but it doesn't specify being a male like it does in humans. So if you start out as a female, that's XX, but during early development, one of the cells loses an X chromosome and becomes XO, you will become a male. Because in fruit flies, amazingly enough, each cell individually counts the chromosomes, the X's versus the autosomes, to determine whether it's a male or a female. So cells that are XO develop male characteristics in those tissues. In this case, they're showing it with some mutations. So we know that the white eye mutation is on the X chromosome. That's what Thomas Hunt Morgan used to figure out X linkage. So if this eye develops from some of the cells that had lost an X chromosome, it will have, and this was heterozygous, like X plus and X white, then you can get a white eye where you get a red one here. And it depends on where in the organism they are. Now they have this one split right down the middle, so the left half is female and the right half is male. That's really unlikely to occur, but it's just showing what it would be. It was like which tissues develop as that. So here again, there's like a mutation that makes miniature wings that do that. This, they can't draw the, they can't draw it because they've got it all in one thing. But you can see here on the female, you get stripes on the end of the abdomen, and there's just a little dark colored, the very tip there. And in the female, see, I don't know if this is drawn correctly or not. I know the male has it's all black there, and then I'm losing it because it's been a long time since I worked with fruit flies, but one of the sexes has this sort of pointy shape here, and the other sex is going to have a somewhat shorter and like bullet-shaped rounded abdomen. And I'm thinking it's the male that has the bullet-shaped abdomen and the female has this longer one, but I'm not really sure. In any case, this allows you to show any recessive alleles that are on the X chromosome. And you can get those weird individuals like the cardinals that you've seen the pictures of, where remember they do ZZ and ZW, and so the female is the heterogametic sex there. All right, oh yeah, gonadromorph, I didn't say the name. All right, so now we go on to the third topic. So we've talked about chromosome rearrangements. We've talked about aneuploidy, in which you get either too many or too few of one or a small number of chromosomes, but you're going to be normal number diploid usually for the rest. In polyploidy, we're going to talk about addition or deletion of complete chromosome sets. So you'll have an entire extra haploid set, for example. Right? So the way that polyploidy usually occurs is when you have non-disjunction due to failed cytokinesis. So if you're talking about mitosis, in an early embryonic cell, here's 2N, replication, now they're replicated chromosomes. They will line up in a straight line on the metaphase plate during meiosis metaphase. Then in anaphase, the sister chromatids are segregating normally, but then something happens. And during telophase, cytokinesis does not happen, and instead of becoming two cells, that cell stays one, and so now you have gone from 2N to 4N. You've doubled the number. Remember, in mitosis anaphase, here you have 4 chromosomes. Briefly, you have 8 because we're counting the chromatids, but because you don't get division into 2 cells, now you still have 8 chromosomes in 1 cell, and so you've doubled everything up. Instead of having 2 of every chromosome, now you have 4. Here's 2, here's 4 in that cell. This can also happen in meiosis. So here you see diploid undergoes replication. Again, these two are identical to this point. Meiosis 1, you get non-disjunction due to the cell not dividing into 2 daughter cells, right? There's not 2 daughter cells and all the chromosomes go to one and none go to the other. This is every chromosome in the genome. So you don't get cell division and you get 1 daughter cell instead of 2. At meiosis II, the cell doesn't know that anything is wrong. So the chromosomes line up in a single file line, sister chromatids separate, and you end up with your gametes here. But instead of being N like they normally would, they're 2N, because the reduction division, which normally would mean you go from having 2 of each, or 2N here, and you should have 2 cells after meiosis I with just 1 N or N each. That didn't happen. So here comes the weird thing, because you're 2N over here, and now you get fertilization with a normal gamete, which is N, and that's how you get a triploid. So you get a tetraploid by doubling up early during embryogenesis, and that cell being going to the germline, and therefore all the other progeny will be fine. Or here you end up getting a triploid. As we'll see coming up, this is going to be okay for meiosis because you have an even number of chromosomes. This is not. Right? So if you're triploid or have 5 or 7 or 9 copies of each chromosome, you're going to be in trouble during meiosis, because what happens at metaphase of meiosis I, the homologous chromosomes pair up. And if you have an odd number, there's going to be, a third wheel or a fifth wheel or whatever you're talking about, a chromosome that doesn't get a pair. And here you're going to have the blue one goes to one cell, and then the other 2 go to the other cell. Now this one's going to be fine. Gametes are going to have just N there, and plus N is going to be 2N. Here you're going to have 2N plus N with the gamete 3N. But you can see that you can have other pairings here that do other weird stuff. And you might be thinking, okay, look at this guy here and this guy here, right? They both have N. So why wouldn't their progeny be normal? Because they have N plus another N, that's 2N, right? Okay. This is just one chromosome. You have 3 of every single chromosome. What's the chances that this cell got one of this set of 3 and also gets one of all the other sets of 3 and not 2? It's pretty low, because all these other methods of pairing and segregating the chromosomes can occur. So what you end up with is gametes that are 3N for some chromosomes, 2N for other chromosomes, and N for some other chromosomes. So it's a complete mix. And then you get 1N or N from each of the chromosomes as a gamete, and those fail to develop, right? So many of you make use of this every time you go to the grocery store. In addition to the bananas listed here, most of the seedless organisms that you get at the grocery store, so grapes and watermelons and so forth, are going to be triploid. Everybody's had a seedless watermelon, right? You don't have the black seed, the big black seeds like you normally get in the regular melon, but what do you got in there? Those little white things, right? And they're not crunchy, and so you can just eat them, so you eat the seedless watermelon. Now, from now on, you could think when you're doing that, you're eating dead watermelon embryos. Poor little watermelon babies that died before they got to become a seed. And the reason is because they all do this, and they don't develop to that seed stage. But occasionally, maybe not in every melon, but every so often, you do see a black seed, don't you, right? And you're like, oh man, because you miss it and you crunch it or whatever. That's the rare case in which one really did just get one of everything. It won the lottery. And so then it got fertilized, and you get one escaper there. All right. So the grocery store bananas. When you bite into a banana and you look down the long way and see the circle, those little black dots in the middle, those are the seeds, but again, they're so small and dead that you can just eat it straight. The wild banana that the ones we use come from have big old seeds in them. Like the white part, the fleshy part of the banana is a lot smaller, and the seeds are a lot bigger to the point where it wouldn't even be worth eating. And I think actually it would be, bananas and plantains are almost the same thing, and so it'd be more like a plantain anyway, where you wouldn't be able to eat it without cooking it. You know, when you open up if you've ever, there's many different cultures that have the plantain as part of their cuisine, but in all these cases, you never would eat one, just peel it and eat it like a banana. You have to like fry them up or cook them in some other way to eat them, because they're not soft. You have to cook them up first. All right, so most store bananas are triploids. We're going to talk about what this means again in a second, but an autotriploid just means it's 3 of the exact same genome. So they are 3n equals 33. That means how many different kinds of chromosomes do they have? 11. And how many of each are there? 3. So there's 3 sets of 11 different chromosomes. All right, so based on what we just said here, why are they not fertile? Why do you get those little black dots that you can eat versus, you know, big seeds that you would have to carefully separate and spit out? What? So let's go through them and say, like, you know, true or false and why for each one. So let's go back and look at this first. What's going on here? What did I say is wrong when you have 3 or 5 or 7? There's not an even number of chromosomes, so what happens at meiosis 1? They don't segregate properly. You get the number of even ones pair up, and there's always one left over. So if there's 3, you get 1 pair and then 1 left over. If it's 5, you would get 2 pairs, that's 4, and 1 left over. If it's 7, you would get 3 pairs and 1 left over, etc. So is there any non-disjunction happening here? No, it's just that the segregation is incorrect because the normal method by which the cell determines who goes where doesn't work. With mitosis, you have 1 chromosome and it divides into 2 parts, the chromatids that get separated. In meiosis 1, you have 2 replicated chromosomes that separate after being in a pair. Here, you can't do that, so the answer is B. So it's not that it has no homologous pairs. For a triploid, you would have one homologous pair for every single chromosome type, and then you would have 1 left over. That's the problem. You're not getting non-disjunction during meiosis 2. You're getting problems with segregation in meiosis 1, that weird stuff goes to the first daughter cell. But then in meiosis 2, in all these cases, everybody lines up in a single file line, and each chromatid or each chromosome separates into the 2 sister chromatids, and everything's fine with that. Because you don't need pairs to make that work. Each chromosome functions as its own. All right, so because you're counting 1, 2, 3, 4, 5, 6. Well, actually, if they were all there, these 2 would both work. But these two are fine for this chromosome, but they're probably not fine for the other ones. So the banana has 11 different chromosomes. They would be fine for one of them, but the chances that they would have gotten lucky like this for all 11, really low. Nope. So here, for all of these, this is only one chromosome. This isn't 3N for every chromosome. This is just 3N for this chromosome. So these cells would be 3N for this chromosome, they would be 2N for some of the other ones, and they would be N for some of the other ones. So every one of these, every one of these could have 0, 1, 2, or 3 for each of the different chromosomes, and it's a mixture. It could be different for each chromosome. The chances that you would get all 3s and then you just add one whole set and make 4, that's a tetraploid, that would be fine, is really low, like to the point of, you know, not. This, you mean? Or making a triploid? So, but that's because we picked the triploid banana, right? We selected for the triploid banana. So the triploid banana, all the cultured bananas do this for all of their gametes. The way that, so, and this is a problem genetically too, because around the world, and read the thing at the beginning of the chapter, it's interesting, all the bananas are genetically identical right now that we have. And so we're in trouble. Because if anything happens, like a new fungus or some other bacterial disease, or anything happens, the population is all exactly the same genetically. There's no genetic diversity whatsoever. And the reason is because we got a banana that everybody really likes, and they're not looking for the very rare seed that forms from this. They're doing what's called plant tissue culture to make the new bananas. You can take a piece of a leaf or a piece of a stem and you treat it with hormones and make it root in usually an agar-based medium, and you're making like little baby bananas that are they're genetic clones of the parent. The way you get the triploid to start with is, as I'll show coming up, is you get a tetraploid and made it to a diploid, because this would have, the gametes here would have 2 each, and you made it to a diploid, that gamete has 1 each, then you end up with 3. See how that works? It's or exactly like this, you get 2N and 1N. All right. So, autopolyploids, they're going to have multiple copies of their own genome, like same species genome. Allopolyploids are derived from a combination of species. And this is where it gets a little bit weird, because you have different species whose genomes are close, and because they're close enough evolutionary relatives that their genomes are compatible with each other. So if you take an allopolyploid, generally these are more vigorous than the individual organisms from which they're derived. So a lot of allopolyploids are developed in the agricultural field. So you can get more robust plants that do more of what you want by combining different species. The bread that you eat, we'll see in the very last example at the end today, the flour that you get, for bread is a combination of 3 different grass genomes. And there's some old, like if you go to the Whole Foods or whatever, there are some of the heirloom varieties, I guess, you'll recognize the names from some of the things we talk, some of the species we talk about at the end here. So these are all, so B stands for Brassica. These are all in the same family as like broccoli and cauliflower and stuff. Yes. It is exactly the same idea as hybrid vigor, except that hybrid vigor is when you cross two plants of the same species, and you're not increasing the number of chromosomes, you're just mixing up the alleles. Hybrid vigor comes from an increased diversity of genetic alleles when you cross two different members of the same species. Here, as we'll see, you're adding one complete genome to a different one. And with hybrid vigor, the two species have exactly the same genes on exactly arranged the same on the same chromosomes. And let's look and see what we got here. This is where this is different. So here are two sunflower species. They're both 2n equals 6. And they both have roughly the same genes, but they're organized differently. You can see on the left, there's like chromosomes A, B, and C right here. And you can see here, this one has G, H, and I. These have speciated, and the organization of genes on their chromosomes are different. So the two homologs of A have all the same genes from one end to the other. There just could be a different allele here than here. But these, although they have all the same genes, they're organized differently in different orders. You can see the chromosomes are even different shapes. So it's not even like all the genes here in that order would be down here in that order. They're all mixed up, such that A compare with A, B with B, C with C, but none of these compare with any of these because pairing is by DNA homology. That's sequence similarity, right? So this species 2 has all the same genes as species 1, but they're organized differently on the carriers of the genome, which is the chromosomes. But they are close enough that if you get gametes from each of these, they are able to come together to make a zygote, and they grow up to make a plant that appears normal. This gets a little confusing. This plant is 2n equals 6 genetically. The plant is genetically diploid. It's got 2 of each of the genes. That's because one of each of the genes is on chromosomes A, B, and C. And a second copy of each of the genes is on G, H, and I. But although it is genetically diploid, we say it is functionally haploid because none of these chromosomes can pair with each other. So this plant, although it's genetically 2n, will never make viable offspring because its gametes will all be just weird mixes of all the chromosomes. None of them can pair, so just random weird stuff happens during meiosis 1 anyway. However, we can fix this. If this plant during an early mitotic cell division has had a non-disjunction event, meaning that if you remember back when I showed you the mitosis slide, so you undergo what looks like normal mitosis. The chromosomes that are replicated at anaphase separate into sister chromatids. Here's one set, here's the other set over here, but then the cell doesn't divide. That will double the number of chromosomes. So you go from 2n equals 6 to 4n equals 12. So you have 4 of each of the genes. two of each of the genes, 2 copies of each, are here on the A's, B's, and C's, and the other 2 are on the G's, H's, and I's. But now, because you have 2 of each kind of chromosome, you can get pairing at meiosis 1 that is just fine. So this plant is This plant is genetically tetraploid because it's got 4 of each gene, but it's functionally diploid because there's only 2 of each homologous chromosome. One diploid set, or one diploid of the genome, 2N, is over here on these carriers, and the other one is over here on these carriers. Right? Yeah. And what? Yes, exactly. Exactly. So, so how do you get a mule? A donkey and a horse, right? Nope. So no. The reason is because in almost all animals that I know of, being tetraploid is not tolerated. There are some examples. So it's theoretically possible, but it wouldn't work. There's one example that I know of, and that is the zebrafish. It is not tetraploid. It is genetically tetraploid but functionally diploid, but it's not an allopolyploid coming from 2 different species. At some point in the past, the zebrafish genome duplicated, so it's got 4 of each gene within its genome, but the chromosomes, there's only 2 copies of each. The other genome is dispersed, throughout the chromosomes. So here's the deal. This is a 2n equals 6, and there's another 2n equals 6. So I can't remember which has more and which has fewer. But I think with the donkey, or with the mule, one of the horse or the donkey has 2 extra chromosomes. So what ends up happening is that you get an organism that has it's haploid for a mule is haploid for horse and haploid for donkey, right? So it's only got 1 of each chromosome. It's functionally diploid, or it's genetically diploid but functionally haploid. This is, if you get an older, this isn't the book that talks about Blue Moon, is it? All right. So there's an older version of this book. So yeah, mules are sterile, right? But amazingly, love is in the air, and people have found, there'll be a mule in a pasture, and I think it's usually with a donkey, but then another donkey the mule will become pregnant, and there will be a donkey. Once in, they named the baby Blue Moon because, once in a blue moon, it's really rare. The reason is because every once in a while, the egg or the sperm from a mule will get really, really lucky and have only all the horse chromosomes or all the... No. It's because of random segregation of the chromosomes during meiosis. Because the mule has only 1 copy of each of the horse chromosomes and 1 copy of each of the donkey chromosomes, there's no pairing. So it's just like whatever goes where is just random. But every once in a while, and there's been multiple occasions, and now, people just thought that another animal snuck in there or something, but they've genetically tested them, and there's multiple examples now. So it can happen because there's lots of eggs produced, it can actually happen. And you get lucky. Like finding the black seed in the watermelon, that got lucky and got to develop into a seed. All right, so now this has been doubled up And so this plant is an allotetraploid, and it is fertile. It makes offspring that are just fine. The gametes are all have one of each here. So really the gametes are diploid, but there's no, every one of the 6 chromosomes is different. All right. So how do we do this? How do we generate triploids? In order to get a triploid, you have to mate a tetraploid, which will produce gametes that are 2N, with a diploid, which will produce gametes that are N. So first we have to get a tetraploid. So Arabidopsis thaliana is a plant with a very fast generation time. It's the plant people's model organism of choice, and a lot of really good work has been done with it. It is a member of the Brassica family, so again, like broccoli and cauliflower and things like that, cabbage. The rosette of leaves is only about that big, and it's very low to the ground, and it makes a flower stem that's maybe about a foot tall or so. Makes lots of progeny, and importantly, it's able to be grown under standard laboratory conditions. So just like the incubator we grow the C. elegans worms in, you can have an incubator that has standard heat, and unlike ours, it will be a humidity control, and also it grows really fine with regular fluorescent lighting. So you can grow them in an incubation cabinet that's not like growing things out in the field where it's like, did it rain today? How bright was the sun? Problems like that. So you take seeds in the first generation, put them into a petri dish, let them sprout, then take the seedlings, and here's where what happened by accident here we do on purpose. So we treat these with a drug called colchicine. Colchicine will inhibit cell division. So we take these early shoots, seedling shoots, that are where the cells are dividing rapidly, and treat them with colchicine for, I think this is like 4 hours or so. You don't want to treat them for much longer than that, because if you inhibit cell division, they will eventually die. And you hope that during that time you get some events where you get a non-disjunction, where you get a mitosis that happened, but then the cells don't divide. Take all these plants and you transplant them. These plants are referred to as chimeric. You can think of this as like the P1 generation, not the F1 generation, not the C1 generation. You plant these and let them grow. These are chimeric in that early on, some of the cells had this event where they went from being a diploid to a tetraploid, but many did not. So some parts of the plants are diploid, some parts of the plants are tetraploid, and what you're looking for are plants in which the parts up here that are making the gametes were the tetraploid ones. Okay? You take all these guys and you plant them. In most cases, you will get diploid plants. But in some cases, you'll get tetraploid plants that have 4 of each chromosome. And these plants are completely tetraploid because they came from cells, they came from a plant here where the gametes were developed from the tetraploid cells. So the gametes, and these are self-fertilizing plants, so the gametes are tetraploid, the plant is tetraploid, somatic cells. The gametes are diploid, so a diploid comes together and makes a tetraploid. Because this plant is tetraploid, it's got 4 copies of everything. That's 2 pairs. So it is completely fertile. So now you can maintain this without any further treatment. You have a tetraploid line and a diploid line of the same plant. Then to get a triploid from that, all you have to do is cross the tetraploid which makes gametes that have 2 of each chromosome, to the diploid, which makes gametes that have only 1 of each chromosome, and then you get 3. So I'd imagine how this is how it works for the seedless watermelons, right? So because the watermelons are seedless, you can't really take the seeds from them and make another generation. So there are farmers that are growing the seedless watermelons to sell at the grocery store. The seed companies are also contracting with other farmers to grow tetraploid melons and diploid melons that they then cross, and the seeds that develop from those melons are the seeds that you plant to get the triploids. So you're constantly having to grow tetraploids, you're constantly having to grow diploids, both of which make seeds. Then you cross these to get the triploid, which makes plants that are fine, grows the vines, the watermelons come out, but then every time you have to go back to here, every year, in addition to all the farmers growing the melons that come to our store, there's farmers that are growing melons to maintain these both, and also crossing these two to get a good supply of the triploids, the seeds that will be triploids. Does that make sense? Yep. So to get a triploid, you take a diploid, treat it with colchicine, get a tetraploid plant, and then once you have a tetraploid and you're maintaining it, all you got to do is keep crossing it to the diploid plant to get the triploid. With stuff like, see, that's the difference with the watermelons. I doubt that they're doing, technically you could do tissue culture, the plant tissue culture, but that would be really expensive in terms of facilities and then having to transplant the seedlings to do that for an annual crop like watermelons that you have to make tons and tons and tons of. So the bananas are different because when you get a banana tree, it's good for years and years and years. So you're doing tissue culture to make an exact copy, whereas here you're doing it the old-fashioned way. All right, so let's talk about this. So if you look some places, so not this, these are all Triticum species. So Triticum is a genus, and then you have the species. But so here's this wild grass, Triticum cerezi, that you've never heard of. But like I said, if you go to like Whole Foods or somewhere, or if you look up, recipes on a slightly foofy website or whatever, that's the heirloom stuff or whatever, you will find einkorn wheat still available. It's a blast from the past, I guess you would say. These are 2n equals 14, so the genome N is 7 different chromosomes. You cross those together and you get this hybrid that is also 2n equals 14. They didn't draw the whole picture because there's too many chromosomes, but this is just like, look back to the sunflower example. This one is genetically diploid but functionally haploid because there's only 1 of each chromosome. There's 7 chromosomes from the einkorn and 7 chromosomes from the wild grass, so there's 14 total, but they don't match because the arrangement of genes has changed over evolutionary time. Then you take this hybrid, which is going to be sterile, but you early on here, you treat it with colchicine like we've talked about, double it up, and now you have another one, emmer wheat, which is also available in Whole Foods and other the Bob's Red Mill and all those little things you can get at Publix. And you see it's all Triticum here. Another wild grass species you made in. This one's going to be 4n equals 28, so the gametes are going to be 2n equals 14. This one is fertile because it's got 2 of every chromosome now. Made it into this one. And now you've got 3n equals 21. Whoops. Because this one, the gametes here from this one are going to be 2n equals 14, but they're going to be half from each of the species. So now you've got 21, but there's 3 different kinds of chromosomes. So these are genetically triploid, but functionally haploid. So there's 3 complete genomes in there, but each genome is in its own species chromosome arrangement. Okay? And then you double this up to get Triticum avestium. You don't need to know these names, just, you know, the process, you know, how you would do this. So this is 3n equals 21. Colchicine treatment, double it up. So now it is 6n equals 42. That's the wheat that's grown up on the Great Plains. That most bread today is made out of. And there are different cultivars of this as well. This one is 6n equals 42. So it's genetically hexaploid. There's 6 copies of each gene, but it's functionally diploid because they come from 3 different species. There's 2 chromosomes each of 7 different, or of 21 different kinds. So that makes 42 total. And then this can be further selected by, you know, regular genetic breeding for different, you know, for different characteristics. And there's winter wheat and summer wheat and all kinds of stuff. Being from the, who knows how to make biscuits here? Oh my God, just one? Are you kidding? What kind of flour do you use to make biscuits? Whose brand? All right. But so people are always taught, it's like there's summer wheat and there's winter wheat and there's different properties of each one. If you want to make good biscuits, from what I've read, this is why the Southerners can make good biscuits that even the best restaurants up north have a hard time replicating, is because you need to buy the White Lily brand flour, which is a different cultivar that is only, you know, has different properties as far as the softness or the hardness of the wheat, blah, blah, blah. Okay, all right, I'll see you guys at lab today. Otherwise, if you don't have lab today, remember the exam is on Thursday. It's exam 2, so you do not need a Scantron. You do need a calculator that is not your phone. Inactivate your phone before you get in here, and you can bring up to 2 pieces of blank paper for scratch paper, and I'll just have you hold them up and show me at the beginning that you'll turn and ask. So you can bring 2 pieces of paper that, you know, that you can write stuff on, like to do calculations if you want. Make sure to be here at 8:30. Right now, that clock is even farther ahead than